Well, I started off my career as a high school teacher. And I'm sure you also would agree it's the most rewarding thing ever. So I also taught high school all through my PhD and my postdoc, which I actually did here. So I've taught physics, calculus, and AP biology. And today I'm going to tell you about something completely different. But one of the things that I really enjoyed about teaching high school is that you get so immersed with the students and you can become like sort of integrated with the kind of things that they're thinking. And one of the things that really inspired me was challenging them to ask different questions about the information that was in front of them. I'm not trained as a teacher, but as a scientist, you sort of get thrown into the deep end of just doing that, teaching people. And that's basically, even though I run a lab now, in essence, that's what I do every day. I wanted to start off with this quote because I actually agree this is inspiring for us as people who work here at Salk, because it really is about asking questions. And the Salk is actually different than other research institutes and other big institutions like UCSD, because we're really focused on answering very hard questions and understanding the mechanism of how things work. And not to say that other people don't do that, but that is like our fundamental goal here. However, I want to change that question because now we're in like a new era of thinking about things. And like there's been a lot of chatter around using large language models. And I want to just reframe this. The questions are still the most important part. And as Jonas said, the information is out there already. We just have to ask the right questions. And I think that this is the beginning of a new way of learning about using the tools that we have. And this is just a tool. Scientists use it. People in my group are using it. We use it to understand how systems work, because it turns out computers are much better at bringing together lots of information. So I think that I'm reframing Jonas's quote in a slightly different way in my lab. All right, so I'm interested in plants. I love plants. Since I was 13, I decided I wanted to sequence plant genomes. If you've decided you want to rotate through my lab today, then you will be learning about how we do sequencing. So I'm going to tell you about lots of different things today, but generally my lab takes the DNA out of plants and understands that code. The code is like the software of a computer. Cells are like the hardware. We try to understand how those two work together to make these beautiful things. So we have these beautiful woods. We have our forests that you may know about since we're all here in San Diego. These are the giant kelp forests that just are offshore, and they're coming back finally. But I'm sure you're very familiar with these trees, which are only found here in California, the sequoias. So one of the questions that I had when I was growing up is I would wander around and be like, well, these are super big trees. I grew up on the East Coast, so they were big trees. I didn't have these trees, but they were big trees. And I'd be like, so what exactly is going on here? How did they get so big? They are pretty big if you put them into the context of other things. This is actually a tree that my lab works on, baobab. This is actually one of the oldest non-clonal plants on Earth, even older than the sequoias. They're just not as big, although they are quite massive. This is the kind of tree that I grew up looking at thinking, how did this actually get that big? So the question really is, where does all that mass come from? And I've only taken one teaching class ever in my life. And that was actually when I was getting ready for grad school, I thought it'd be fun to take a class. And actually, this question came up in the class, and you might be familiar with the study that you can find online. And if you haven't seen it, just Google where did the mass come from, Harvard, MIT. Super fun. But the question is, like, where did this mass come from? Because when you look at these are actually seeds from the redwood. They're tiny. So the plant starts off with this tiny little seed and then grows into this massive tree. And I'm left asking, where does the mass come from? So where does the mass come from? You're smiling, so you know the answer. carbon dioxide, and sunlight. And if you are interested and pull up that video online, you'll see that a lot of people think, oh, well, maybe it came from the soil. And the thing is, this is the kind of abstract question and then answer that really gets people excited about the things around them. Here at the Salk, we thought, okay, this is actually pretty cool. Look at this. Plants are sucking in carbon dioxide, making mass, and releasing oxygen for us. And they do this through this fantastic process called photosynthesis. It requires light, the sun. Well, you could have artificial light too. And this process has a very specific equation. So we can basically work from carbon dioxide dies X, we can observe it, but then we can actually start to break down and ask questions. How does this actually go? What's the stoichiometry of this situation? These kind of questions then can lead into very complex ideas of how things actually work, like how much sun is required, how much water is required, what does actually glucose, how does it turn into the mass of the tree, like lignin? One of the questions that I think is the most exciting part of this and make plants like truly phenomenal is the answer to this question. Does anybody know the answer to this question? You can't answer, Carl. You can't answer, okay? No. What is the most abundant protein on it? I just told you that plants are so important. What exactly are they doing when they capture carbon? Well, then everybody's going to learn something new today. This is super fun. Rubisco. So, the most abundant protein on Earth is the protein that actually captures the carbon. Which, I guess, now, it's like, oh, that's obvious. But this is the protein that we now are becoming very familiar with. So it turns out that Rubisco is a great protein for us to eat because we're used to digesting it, because most people at some time in their life actually eat leaves off of plants. And guess where those proteins are? They're in leaves. So you have ways of digesting it. The problem is that a lot of these protein-based foods are actually made out of non-Rubisco proteins. I'm not advocating one way or the other. I'm just stating that that's what you're eating. And I'm going to tell you a little story about my friend here later. Yes, you're going to get to take some fun stuff home. And I want you to think about that in terms of like asking yourself the question, would it be better to eat a protein that we already digest well, or should we eat proteins that we don't digest very well? All right, so plants, I hope you are impressed that they are the lungs of the world. And we know this because we didn't exist until the plants came about. This is a great paper that was in the Proceedings of the National Academy of Science that basically shows the mass on the Earth. Yes, this big green thing right here, this is the plants. This little box here, this is us. Oh, wait, no, this is us. The little box. We make up a very small portion of the proteins in organisms on Earth. If we didn't have this, we wouldn't be able to breathe. And usually I show another slide that actually shows before plants, there weren't anything else. So we have a very tight relationship with plants. Now, we see plants and we think, oh, they're so big and beautiful, but we don't think too much about what's underground. And that's what we're very interested in here. We started asking questions like, should we think about the roots more? Like, as we're thinking about drawing down carbon, what's the role of the roots in that process? Most of the time, you see very little of the roots. I mean, sometimes you see some, like especially when they're eroded away, but generally, most plants, non-crop plants, have the same amount of root structure underneath of them as above. So that's a lot of biomass right there. And it turns out that all of this beautiful humus soil that we have in the Midwest is actually the result of prairie grasses and other prairie plants leaving behind the remnants of their very, very deep roots in a place where bacteria cannot degrade them. The oxygen stops around here. And then all of these roots will break off, and they leave behind remnants that over time create the great humus soil that we're familiar with in the Midwest. Now, it would be great if we had some of that here in Southern California, but we have plants that actually don't need great humus soil, like sorghum, or better known as Sudan grass here. So we started asking questions. Can we actually leverage plants to help mitigate climate change? Draw down carbon. And this program started, it's called the Harnessing Plants Initiative, and we have support from the Hess Corporation, Bezos Earth Fund, the Audacious Project, and then HHMI. And it's made up of the plant professors here at the Salk. It's led by Wolfgang Busch and Joanne Corey. And basically what we're doing is we're trying to make plants that have deeper roots. Because over the last 100 years, we've done a really good job of breeding better crops to make more yield. But we did that mostly by putting more nitrogen, phosphorus, and phosphate on the soil itself, making it so that deeper roots aren't really necessary anymore. So a lot of the current crops that we grow have very shallow roots. And so our thesis is that we can just go back into some of these crops, do some more breeding, bring back the larger roots, and then you can imagine if we utilized the entire agricultural system and put a little bit more carbon back in the soil, maybe we could capture more carbon, but we could also possibly increase the quality of the soil. So we're doing this both in current use soils as well as soils that haven't been used or fallow because they are low quality. So the Harnessing Plants Initiative has 3 projects underneath the great umbrella. Crops is the one I just told you about. We're leveraging the scale of the agricultural system to just put a little bit more carbon back in the soil, improve the soil. We have CPR, or coastal plant restoration, which I'm not going to tell you about today, but the idea there is that wetlands actually store 30% more carbon than dry land. And we're losing a lot of our coastal areas, because we like to live near the coast, right? But a lot of those coastal areas are now being restored, and so we are using genomics to say, yes, this individual fits better in this wetland. So we're matching genotype to environment. So there is a lot of restoration that already goes on, but a lot of those fail because you pick the wrong plants to go in those locations. But I want to tell you a little bit about plants of the future. Enter in our friend today. So I know that you're all anxious to get one. So please, if you could, take one and pass it around. And you may already know what this is, but maybe you don't. And I'm going to tell you. Okay, so there's this awesome paper that came out in Nature. Nature generally is considered one of our top journals in science. So if you're ever interested in actually finding out what good science actually is, Science, Nature, Cell. For us, Plant Physiology, Plant Cell, these are all the journals that we read. This paper came out in Nature in 2006 describing the Azala effect. Azala is actually a fern, a very tiny fern, as you can see, and it fixes its own nitrogen. So it has a symbiont, cyanobacteria symbiont, that actually helps it fix its own nitrogen. It is a freshwater plant, and the idea here is that it grew out of control in one of the lakes on top of the earth and basically took over that whole lake. It sucked so much carbon dioxide out of the atmosphere, we had a change in the climate, it all sunk to the bottom. Some people think this is actually what we're driving our cars on right now. Because plants turn into oil when they decay. So with this inspiration, actually around 2006, but I was also very familiar with this system already, I decided that I was no longer going to work on our favorite model system, which is Arabidopsis thaliana, and I was going to work on my new favorite organism, which, by the way, was the model organism before Arabidopsis, so I was basically just going back to the old model organism, which is duckweed. So what you have in your hand here is Lemna menuda, specifically collected from Copper Creek Park in Encinitas. And if you've ever been to Copper Creek Park, it's basically like a sewer. So this, I've grown this like many, many generations, so it's not good. Okay, just so. So this has been sterilized. So right now, the plants that you have are sterile, or they should be. So I've taken this many generations. We actually have a paper describing the genome. This is duckweed. Duckweed is made up of 5 genera. We have Spiridella, Landoltia, and they make up these like more leaf frond kind of things. We have Lemna, which is the one that you have. Then we have the smaller ones. We have Wolfiella, we have Wolfiella, and then I'm going to tell you more about another Wolfia, because Wolfia is actually the smallest plant in the duckweed family. Duckweeds grow everywhere, so this is actually 2 plants that pretty much grow everywhere in the world. Typha and duckweed, and a lot of times they grow together. Anytime that there's nutrients that are sort of flowing into water, like runoff from our yards and things like that, either duckweed or typha are going to suck up those nutrients. Now, if you go out today and actually start looking around for typha, you'll see it everywhere. Duckweed, you have to look a little bit more closely, and around here it actually starts to come out more like April, May time, because it actually overwinters underwater. So the duckweed that we're working on is actually called Wolfia australiana, and it's the size of a pinhead. This is basically the entire plant, and it buds off like yeast. So basically when it grows, it's basically budding off new mother fronds that are daughter fronds. Recently, we did what we call a single cell experiment, and we figured out that it only has 4 types of cells, and then a couple of subtypes. And this makes it ideal to actually start asking questions specifically about how the plant processes carbon dioxide. How does it actually grow? These are the kind of things that make it an ideal model. This is some really nice microscopy done by my collaborator Kevin Cox at Danforth Center, basically looking at the different features of duckweed. This is actually Wolfia microscopica. It is one of the ones that we get to flower. So this isn't just something that we're doing in the lab, though. This is actually something that has been part of the human experience for a long time. It's the smallest and fastest growing plant on Earth. It also makes the smallest flower. So this is actually Kelly Colt in my lab took this picture under one of our microscopes, and basically it flowers all the time and makes seed. It's beautiful. So one of the reasons why they chose Arabidopsis thaliana to be the model plant over duckweed is that it's much easier to get seed and do genetics from Arabidopsis. However, we have created a system where you can actually easily get seed, and instead of getting like seed from one plant, you get seed from a whole population, but they have the same genome, so the seed actually breed true. Now, this is related to another plant that you may be familiar with. At the San Diego Botanic Garden, we have the corpse flower, Amorphalis, and it basically is in the same family. So the largest flower and the smallest flower. Plants are amazing, aren't they? Just doing so much cool stuff. And then this is actually Wolfia australiana growing. It actually grows and multiplies or doubles in less than a day, 16 hours. So it's the fastest growing plant on Earth. If you give it unlimited nutrients, it will fill an entire body of water. So this is actually a picture from outer space from NASA of a lake where lemna, one of the lemna species, got enough nutrients and it just exploded. So there's a picture of before and after. I guess I should add the before so you can see that there's nothing, and then like a week later, it's full. It's crazy. But wolffia has always been something that we've known was important because in many cultures, it's actually eaten for its protein. So this is actually in Southeast Asia, it's still eaten predominantly for protein. It's 35% protein, which protein is that? Rubisco, exactly. So there's this company, Green Onyx, in Israel that is making, or at least, I don't know if they're still making it, but I just still love this picture. They say they're making it. Like, why not have wolffia for breakfast? It's like little pieces of green caviar. So how could this help with climate change? So I would say we need to refactor the way we think about things, right? So if we think about the current scheme, well, duckweed may not be great just on its own collecting carbon dioxide. But if you think about if you use duckweed, what it replaced, then you could actually start thinking, okay, well, to do this, there's a lot of fuel that goes into making this. There's actually a lot of inputs, nitrogen, phosphorus, phosphate. Then you have this, and there's a lot of resources that go into this to then get transported to In-N-Out Burger so you can have it. But imagine if you computed all of those costs and then compared them to growing them in your home, which I don't know if vertical farming is going to be a thing that we do in our home anytime soon, but you can imagine a future where maybe you are doing that. It's much more practical than this whole like Green Onyx like coffee machine, like I'm just going to dispense a little bit of duckweed for myself. I don't know if that actually would work, but this actually does work. So my colleague Eric Lamb at Rutgers University is building these systems and promoting this because it is sort of yummy. I wouldn't suggest eating this one. This one, and it's not because I collected it from basically the sewer, but because it doesn't taste good, right? So wolffia tastes a lot better than lemon. But I wanted to give you a line that was local and not one that was not local, because just in case it goes somewhere. But this is one where you could leverage it and start having your students ask questions about plants. Like, maybe they're like, that's not a plant, that's bacteria, or that's algae. No, that's actually a plant. Like, plants have many different forms. So duckweed, I think, has the potential to change our future. And I'll just end by saying plants. So some questions?
We have time for a couple of questions.
How do we grow them in the classroom?
Well, that's the great thing about duckweed, is that technically you could take that, turn it upside down, put it on your windowsill, and my bet is that in a month it will be growing, it'll be full. So you don't have to do much. If you were going to try to grow it with your students, then Miracle-Gro, just put a little bit of Miracle-Gro in water, put that in water and some light, and it should go crazy. I mean, that one specifically it grows. If you give it a little nutrients, it's got a taste for nutrients, you know what I mean?
Yeah, thank you.
When you were talking about having the roots grow further to help with the amount of carbon, would it impact how long it would take for those plants to mature versus how they are now?
Well, what we're aiming to do is actually not change any of the maturity of the plants. Specifically for crops, we can't delay or advance the timing of the harvest at all. So making more roots shouldn't actually impact that.
Okay, thank you.
Are you setting too
so we had another person who's actually very, very new to the Salk, just opened a lab here, and she's joining our HPI team, and she's looking at mycorrhizal fungi to see how it actually interacts with the roots. You know, what are the roots actually making that attract the fungi, and then also how do they actually, how can we use that to bolster the roots and make them bigger?
My students are working on exploring plant-based protein, foods, and there's so many little tiny startups around the world. Where do you see in the future, broader question, being successful?
Well, I'm a little bit biased. There's a company here in San Diego that actually is using duckweed for plant-based protein. And so there have been a lot of companies that have come onto the market for using duckweed as a protein source or as to make pharmaceuticals. And many of those companies have failed because they hadn't come up with really good ways of growing duckweed. So when you grow it to make that much protein, you need to be able to grow it at scale. And it turns out that it has some of the same problems that algae has, that when you grow it at scale, it will actually crash or just die all of a sudden. So this company called Plantable, which is actually out in San Marcos, they have impressed me that they can actually grow it at scale, and they've come up with a very cheap way of harvesting it, and the protein is so high quality. What is the protein again? Rubisco. The protein is so high quality that you can take the protein and actually whip it up into a meringue. So duckweed protein is really high quality, and so they're thinking about using it as a filler against other proteins that are maybe have a little bit lower quality but are cheaper. There's another nested question about the protein-based market right now that's different, but the really cool thing about using duckweed is pretty much you can grow it anywhere, right? So then you could have protein sources anywhere, which is not true about the other type of protein sources. The bad thing about plant-based protein right now is the market's completely dropped out, which has actually made it difficult for a lot of the companies that are currently in the market to compete. But the primary protein base is, soy still. So.
Could you repeat what kind of species this was again?
Lemna minuta.
Okay.
So it's minuta because it's actually one of the smallest of the lemnas. And it's not invasive here in Southern California, but in other places of the world, like Europe, it's invasive. Because it's just so good at growing. Yes, sir?
Yeah, if you're using duckweed, right, to draw carbon out of the atmosphere, where are you then going to put the duckweed? You're going to bury it under the ground? How do you foresee that part of the problem?
Yeah, see, I don't think that is the best solution, like burying duckweed, although that could be, we get these lakes that explode. We could come up with a way to make them all go in. Okay, so let me tell you one other part of the biology. I should probably. So I mentioned that you won't see duckweed right now because it overwinters, or actually will sink to the bottom of the water, and it does that by a process known as terrain formation. So it's similar to a seed, but is actually distinct because it's actually still the full plant, but it's turned its protein, 30% protein, into 30% starch. And that starch makes it heavy and it sinks to the bottom. And so those will sense the change in temperature and light, and then they will germinate. But what really happens is that a daughter frond that's not a terrain will emerge from the terrain itself. So in essence, we could trigger a terrain event, fill a lake and then trigger a terrain event and have them all sink to the bottom. And if it's at the wrong time of year, then they probably would die and you have all that carbon at the bottom. However, I was more suggesting that I think in the future scenario, it's something like this. It's not about what carbon will actually be captured specifically by duckweed, but by the carbon that is offset by not following these other paths. So I don't think that in the same way I described our crops project, where we're leveraging the agricultural scale to put more carbon in the soil, I don't think that duckweed or these other species will play that role. They could, I mean, like, and we could think of ways of doing it. I should have also mentioned that we're working very closely with other organizations like NASA and other space agencies that are interested in duck meat as something that you will eat, not only in the space station, but on Mars or whatever planet we go to.
How, can they grow, are they, like, can they be extremophiles at all? Can they grow in like really salty places?
Well, they are sensitive to salt, but they do grow pretty close to salt, so you can get them up to a certain salt level. They absolutely require water. So if you don't have water, so the desert, they don't mind heat, and they also don't mind a lot of heavy metals. So in many places in the world, China, Australia, they're using it to actually remove heavy metals from water. And, you know, going back to Copper Creek, I think it's actually removing a lot of stuff before it flows into the ocean at Moonlight Beach, so hopefully. Thank you.